Hi everyone. This week we'll be looking at chapter 4 of the book. We'll be looking at radiosensitivity and the response of cells and tissues to radiation. In particular, we'll be looking at the shapes of survival curves for acutely responding and late responding tissues. So in terms of the tissues and the organs, the killing and depletion of the critical target cells in a tissue is really what is responsible for the visible and detectable changes induced by the radiation. And the sensitivity can be defined as the loss of the reproductive capacity for these cells and depend on the inherent sensitivity of the various cell populations in the tissue or organ. And also the turnover kinetics of each population in the tissue. In other words, how often do they divide? Now, these two guys are Bergeny and Tribondeau, and they basically deduced that cell division played an important role in radiosensitivity based on reports that X-rays appeared to destroy the cells of a malignant neoplasm, a tumor, without permanently harming the adjacent healthy tissue. And so their hypothesis was that ionizing radiation is more effective against cells that are actively dividing or undifferentiated. Now, to test this, they radiated rodent testicles because the testes contain mature cells, the spermatozoa, which contain the male's genetic information, and also contains immature cells, the spermatogonia and the spermatocytes, which have no function other than to develop into the mature functional spermatozoa cells. That said, though, this is more of a rule of thumb than an actual law. And other people have sort of challenged it. So in 1925, Ansel and Wittenberger modified this hypothesis by proposing that the inherent susceptibility of any cell is the same. So it's not the cell being damaged which is different, but it's the time of the appearance of the radiation-induced damage that is different among the different cells. And they found the appearance of radiation damage is influenced by the biological stress on the cell, the necessity to divide, and the conditions to which the cell is exposed. And what was interesting is they found this even for post-irradiation conditions that, back then we didn't really understand, but now we know that's because this can allow for the repair of potentially lethal damage. Now, all cells will be damaged to the same degree by a given dose of radiation. But what their hypothesis is saying that the damage will be expressed only if and when that cell divides. In other words, all cells are equally sensitive to radiation, but more rapidly dividing cells express the damage sooner and appear to be more sensitive. So let's think about what we mean when we talk about differentiated and undifferentiated cells. So differentiation is often used to describe whether cells are mature or not. So a differentiated cell is a mature cell line that performs a specialized function or has some inherent structure in the body. While a undifferentiated cell is an immature cell line whose primary function is basically just to divide. So it's the undifferentiated cells that are both sort of maintaining their own population by constantly dividing, constantly regenerating them. But the other function is to replace mature cells that the mature cells aren't dividing, they're not creating new versions of themselves, but they are lost, right? They are lost from just the regular wear and tear in the body. So, for example, in the testicles, in the testes, spermatozoon is a mature, non-dividing cell that is very specialized, right? In terms of carrying genetic material. And as they are periodically lost, they have to be replaced. And to do this, we replace them by differentiating spermatogonia. That, these are the cells that will mature into those spermatozoa. So we can also divide the cell populations into three categories: stem cells, transit cells, and a static population. Now, the sole purpose of the stem cells is to divide. These undifferentiated cells replace their own population and replace the cells of other populations. And examples might include epidermis cells, at the base of the basal layer. Cells in the bone marrow that are going to go on and create, red and white blood cells, for example. Cells in the crypt of Leverkusen, in the intestines, and spermatogonia, which we just talked about. Transit cell populations are cells that may or may not divide, and are basically they're sort of like I guess transit cells is a good way to describe them. They're sort of in between the stem cells and the more static populations. So they're moving from the stem cell compartment towards this end cell compartment, the static cells. And the static population are fully differentiated and exhibit no or very little detectable mitotic activity. These cells are the ones that are generally lost throughout the lifetime of an organism, and the ones that are going to have to be replaced by the stem cells turning into transit cells, turning into these static cells. Now, in terms of the radiation response to different cells, they can also be categorized in terms of these 5 different cell types. So, the vegetative intermitotic cells, these are ones that are rapidly dividing and are very undifferentiated. The differentiating intermitotic cells are produced by the division of these vegetative intermitotic cells, and although actively mitotic, they are more differentiated than the vegetative intermitotic cells. And then in the middle, and sometimes this class is missing from these tables if you look at different textbooks. But here we have the multi-potential connective tissue cells that's basically intermediate in their radiosensitivity, and include things like endothelial cells, the cells that might line blood vessels, or fibroblasts, the basically the connective tissues. And then the 4th class is reverting post-mitotic cells. Now, these cells typically do not undergo mitosis, but they do retain the capability of undergoing division if it's necessary. And so examples may include liver cells and mature lymphocytes. And then the last one is the fixed post-mitotic cells that do not divide. And because they're highly differentiated, they're not dividing, these are the cells that are going to be the most resistant to radiation. Okay. Now, in terms of the tissue response to radiation, then target cells do not imply that radiation is in any way selective, right? Like we've talked about this in the past. What we're saying is that cells that divide rapidly, like stem cells, are more radiosensitive because damage to those cells, in sufficient numbers of cells to affect it, can be more readily felt, right? But these, because they have a high turnover, they the if you were to destroy these cells, then they wouldn't be able to regenerate their lost populations. But more importantly, they wouldn't be replacing the more mature cells, and so that is really where you will see, the functional and structural tissue damage occurring. Right, it's usually a delayed response from the stem cells being damaged all the way down to when the effects of this is seen. To help sort of understand this, we can look a little bit at the tissue organization. So, tissues and organs are made up of two compartments. The parenchymal compartment, containing the cells characteristic of the individual tissue or organ, and the stromal compartment, which is composed of connective tissue and the vascular tissue, which makes up the supporting structure of the organ. Okay, and just for clarity, the image on this slide is from the esophagus, the food pipe. Now, in terms of how responsive these tissues are, then here's an example for basal cells. So, acutely responding normal tissues manifest radiation damage typically within a few months after irradiation, because they are self-renewal tissues containing rapidly dividing stem cell populations. So, for example, the basal cells produce new skin cells as old ones die, and then damage to these cells would mean the basal cells would not be there, right? If you destroy the basal cells, then they're not able to differentiate and then go on to create new epithelial cells. And so there's a delayed response, in this case of about a few months, where those effects can often be seen. Another example of an acutely responding tissue is the crypt cells. So the crypt cells, these existed within the small intestine at the base of the villus, and provide stem cells for renewal of the intestinal epithelium, which basically turns over every 3 to 4 days, right? Under the harsh conditions of the digestive system. And so radiation effects, we said for acutely responding, they usually seen within a few months. You can actually see the radiation effects much sooner, within a week or 10 days, upon these cells being damaged. Another example would be spermatogonia. We've already seen how the spermatogonia differentiate into spermatocytes and ultimately differentiate into the sperm cells. Irradiation of the spermatogonia would result in the depletion of these cells that are rapidly turning over, right? And ultimately would result in sterility. More acutely responding tissues include bone marrow stem cells. These are very radiosensitive cells. These cells are produced by the bone marrow, the tissue, the spongy tissue found in the center of bones or some bones, and can differentiate into different types of blood cells. So they can differentiate into red blood cells, which carry oxygen around the body, or white blood cells, which help fight infection. And we'll talk more about the effects of radiation on some of these systems later on in the course. In terms of late responding, late responding is actually kind of a bit more contentious, because some people say, well, even though the effects are less pronounced, you still see them. So is it really late responding? But anyway, some tissues are considered late responding and do not express injury for months or even years after radiation. And so examples might include the lungs, the kidneys, the central nervous systems. In these systems, the cells divide more slowly, and they're generally more radio resistant, which we'll talk about later in the course. And this reflects the current philosophy that the radiation response of all tissues results from the depletion of the critical parenchymal cells, right? The cells that are reproducing, and that the difference in time at which early and late responding tissues express the radiation damage is just simply a function of different cell turnover rates. So how long it takes for these cells to divide.